Okay, good afternoon, everyone, and thank you for joining us for this session. I feel in many ways like I'm an interloper here because one of us is not like the other. I don't do CAR T therapies, these two do. Marcela and I will be moderating this session with Carl June. None of them need an introduction. That said, why don't we go ahead and get started since we have a few minutes to talk about CAR T. And you heard a little bit, of course, on this just before this session. So our goal is to ask Carl questions, but I'm going to use this as an opportunity to ask both Marcela and Carl questions. But with that in mind, I'm going to turn to Marcela first to ask the first question.
Thank you so much, Ravi. Carl, we've been in log phase growth in gene and cell therapy, thanks in large part to the efforts from your lab and Penn in the last 10 years. And I'm wondering, what do you see as, how long are we going to stay in that, in the short term and the long term, and what do we need to do to keep that pace up?
Well, you know, I think it's a great question. And in some part, I think you can use a history of natural drug development. There'll be new kinds of cell therapies now to go after the resistance mechanisms that have been identified. And there'll be new kinds of cell therapies that'll target the tumors that yet, have not been successfully targeted. And in the former, I think, the main issue has been, ALL, there's basically a 90% complete response rate. And I think the only issue there is mitigating toxicity, and that clinical teams are getting better and better with experience and modifying dosing and scheduling. In the case of lymphomas, though, it's a 40 or 50% complete response rate if they're refractory lymphomas, and there are efforts to move it up front. And then the main resistance issue has been loss of target. And so I think for sure we're going to see combination therapies, and there are academic trials now testing, in addition to CD19, other targets like CD20 and CD22, and I know you have efforts in that area. And then another issue really has been, we have the huge emergence in 2021 of myeloma with BCMA. So B-cell maturation antigen really, I think, says that we can get the similar overall response rates, 80, 90%, in heavily pretreated patients. So it says that you can change from, the target from CD19 to BCMA, another lineage restricted antigen, and that effector T cells work. I think that's a huge lesson, and that then is going to open the area to solid tumors. But I'm really optimistic that this, quote, logarithmic growth is going to continue because the myeloma showed that it's not a one-trick pony.
Well, I'm going to use what your last comment was to go to the next question, and this was touched on by Ned and David just a few minutes ago, and that is the solid tumor opportunity. We've heard throughout the conference about CAR T and solid tumors, and we heard an opinion just before this, and I'm curious, one, if you heard what they said, and even if you didn't, what is your opinion, both Carl and Marcela, on this point with regards to the potential opportunity for CAR T and solid tumors?
Well, I'm bullish on that. I think the issue is a time frame of what it takes to solve. So there are some scientific issues to uncover the resistance mechanisms. And what I'm positive about is you can take, for instance, an effector T cell and make it a CAR, and take tumor cells from primary cancers that reproducibly kill patients, lung cancer, pancreatic cancer, and in vitro, the CAR T cells will kill the cancer cells. It usually takes a little bit longer than it does with a hematologic malignancy, but they can do that. So there's no cell intrinsic failure if you can get them mono a mono. So there's very complex tumor microenvironment in glioblastomas and pancreatic cancer cells where the CAR T cells may not traffic in, and there's much more challenging microenvironment issues such as the metabolic issues of nutrient depletion, hypoxia, and so on. So, as an optimist, there's a recent study in Nature from Crystal Makel's group at Stanford in a pediatric incurable tumor, diffuse intrapontine gliomas. And They showed that you could give CAR T cells in this tumor and have very promising responses, but they gave the T cells actually intracranially. And so there's one big issue, which is trafficking, and this in part helps solve that. And so I think that's the first foothold really in solid tumors. And then we have had recently both toxicity and efficacy in prostate cancer in elderly men with castrate-resistant prostate cancer and CAR T cells targeting the molecule PSMA. And so it's not yet ready for prime time, but I think that the field is going to solve these scientific challenges in solid tumors. And maybe we're 10 years away. I mean, I think where we are is very similar to where we were back in 2010 when several academic centers had responses with CD19 CAR cells for the very first time. And then, you know, it has evolved to where it is.
Care to comment?
Sure. Of course, I agree with everything Carl said. And to put it in some of the terms Ned was using, I think we're a couple R01s away from solving the solid tumor issues in terms of building appropriate models that would predict efficacy and also that have a tumor microenvironment that recapitulates what is seen in patients. One of our challenges has been that most of the modeling is currently done in immunodeficient mice that don't have a full tumor microenvironment. And at the same time, a fully syngeneic model, although it recapitulates perhaps a tumor microenvironment or neoantigen mutations, it does not recapitulate the human T cell or any human immune cell, really. And so trying to find ways to have hybrids or humanized models or technologies that will allow us to model that. And then I think there also needs to be academic development, and not just R01s, but perhaps, you know, translational sort of studies where we look at the mechanisms of resistance in solid tumors, both in vitro and in vivo, but also then in patient samples. We recently published a story about resistance mechanisms in solid tumors based on an in vitro CRISPR screen. And one of the questions that we get asked a lot is, well, can you do this in patients? And the challenge is that there's not enough sort of centralized data from patients with solid tumors who've been enrolled in a CAR T trial to be able to ask that question. And so if everybody does if there's a very distributed sort of multiple phase ones that are completely designed very differently and that there's no sharing of that data, then it's very difficult to learn what those in vivo in human resistance mechanisms are. So I think that that's an opportunity for us to really work together to figure that out. And working together can take many forms, including, you know, publishing and sharing that kind of information with the world as Penn and community have done.
Great. Thank you. So I have another question, but remember, both of us are moderators, so feel free to ask. and he's the person who's going to answer the question. So okay. I'm going to ask you both again about allogeneic CAR T. Earlier today, Vicky Sato talked about reducing the complexities with regards to CAR T therapies, and in that spirit, thinking about manufacture and having a plug-and-play opportunity for CAR T therapies so that we don't have to have these large, expensive infrastructures and to redo applications over and over again. In that spirit, at least one of the components might be allogeneic, and I'm sure you each have opinions on this particular topic. Carl?
Yeah, well, thanks, Ravi. I think, and also considering back answer to the question, Marcela, about what the next generation of therapies will be, in addition to multi-targeting, I mean, it's going to be using different cell types. I think for sure we'll see engineered natural killer cells, macrophages, and stem cells, and induced pluripotent stem cells. So we'll see different cell types, which will have sometimes overlapping, but other times complementary properties over what a T cell does. And you know, natural killer cells don't, in general, cause graft versus host disease. So for allogeneic cells, I mean, they're really ideally situated and don't require the CRISPR genome editing that you have to do with mature T cells to avoid graft versus host disease. So I think, you know, I don't think we're going to have one or the other. I think we're going to have long-term the ability to make N of 1 T cells, which is going to get more efficient and less vein-to-vein time, but can last for a decade. That's what our recent data shows. But we're going to also have these off-the-shelf cells that can be allogeneic, and they'll have some non-overlapping uses. The immediate use of an allogeneic cell product, and that lower cost of goods is going to be very attractive. But for long-term immunosurveillance, I think that autologous cells are going to be the way to go.
Marcela?
Yeah, I was actually just sort of building on that, since I'm sort of answering questions and moderating, is that, you know, there's all this excitement about these new gene editing tools that don't make double-strand breaks and so on. And at the same time, so there's this sort of scientific excitement, and then there's 2 recent FDA guidances about genome editing and about cell therapies and design. Have you had a chance to look at that? And what do you think that means with this sort of rapid development on the scientific front with new gene editing tools, and also perhaps more regulation coming our way?
Well, you know, in specific, the guidance that Marcela is referring to came out in March, so it's open for comments right now. And one of the guidance was on, you know, an IND really for CAR T cells, and from what I recall, that was 35 or 37 pages long. And then the other one was on genome editing, and it's really a huge step forward for the FDA to now have guidance on these, on what needs to be done. I will say that, you know, for CAR T cell manufacturing, you know, out of the 35 or 37 pages, there was only like 1 page to the allogeneic part. And I think there's and so I think that needs to be expanded on quite a bit. I think that allogeneic cells are off the shelf. I mean, actually will not have the exact same safety profile that autologous cells have. So we've now had, it's close to 10,000 patients worldwide given autologous T cells. That's data from Bruce Levine, who's the president of the International Society of Cell Therapy. And no autologous T cells have transformed. I mean, that's the real Frankenstein thing that people are afraid of. Will this genome editing of the cells create cells then that can then later transform? That has happened now with allogeneic cells. That's not common, but 2 cases were reported last year in Australia where they were allogeneic T cells, and then they transformed and became malignant. We don't know the mechanism of that. So I think this is going to be something, the allogeneic cell therapies, either they're going to need more controls, such as suicide switches, or the understanding of what the safety points really are with allogeneic cells compared to autologous cells. So I think we have now, I mean, the 10,000 patients safety record with autologous cells is huge. If you go back with and look at chemotherapy, cytotoxic chemotherapy that reproducibly induces double-stranded breaks, as you know, there's at least a 5% incidence in the survivors of that, you know, if you look at breast and ovarian cancer, who later get myelodysplastic syndromes or AML, so due to stem cell damage. So I think we can say that autologous T cells with genome engineering with the procedures that have been used are much safer than what we used to have, you know, with exposure to cytotoxic chemotherapy agents. It's still early days with allogeneic products and with other cell types that aren't autologous T cells. So we don't know the safety profile yet of natural killer cells or, for instance, stem cells.
Audience question on allogeneic, which we just addressed. The other audience question that just came in has to do with rapidly progressive liquid tumors like AML. And do we see the opportunity for these kinds of therapies, CAR T therapies, for very rapidly progressive tumors? Marcela?
Sure. I think we're very bullish on, I always get the bull and the bear confused, sorry. We're very excited about the opportunities in AML. I think, you know, between Penn and various other places, we've really optimized and could still optimize some more the autologous manufacturing process. Penn recently published on like a 1-day manufacturing process. That when we've tried 7 days, it turns out that it's difficult to do all the orchestration around getting manufacturing done and released and administering it to the patient. So somewhere between 1 and 2 weeks seems like a reasonable amount of time. And I think the challenge with diseases like AML has been target identification and then managing patients who are very sick and who have a rapidly progressive disease afterwards, and on target off tumor toxicities. But I think all of those are actually manageable, and I'm very optimistic about novel targets and novel technologies that just really improve that autologous space. Kristen Heggie was here yesterday, and she mentioned about if she had to bet on the engineers and solving engineering problems like how do we get automation, how do we get biologistical transport, how do we solve that complexity, she would bet on that over betting on changing the biology. And Pablo Cagnoni mentioned that even sea urchins have MHC, which I did not remember from my immunology class. And so overcoming immune rejection and barriers to allogeneic, I think, is a biologic problem. It's perhaps an easier regulatory path or a drug model sort of path, and so I can see the advantages to that. But I think it's harder to solve that biology than it is to solve the engineering problem of autologous cells. That said, there may be some diseases where allogeneic makes a lot more sense, either because the host has diseased T cells or because or diseased immune effector cells that can be modified, or there's an underlying genetic disease, or there's a non-cancer disease that needs to be replaced with an allogeneic system. So I think there's definitely room for that kind of technology, but I don't see it as something where they necessarily have to compete head-to-head with each other.
Great. Okay, next question, and this has to do with trial design. Peter Marks is going to be talking, I think, later in this meeting from a regulatory standpoint at the FDA, thinking about simplicity in CAR-T and trial designs is going to be one of the topics with regards to cell therapies. Carl, any thoughts in terms of novel trial designs and openness to that, either from a regulatory standpoint as well as execution?
Yeah, so I think one lesson so far, Ravi, with trial design from engineered cell therapies is we're looking for major therapeutic effects, and so that we don't need to have large numbers of patients. And so smaller trials, but very deep analysis of the patients after they've been treated, I think is the way to go for these cell therapies. So we're not looking for marginal effects, but for potent therapeutic effects. And if you don't see that in 10 patients, there's probably time to move on to a different approach. There's so many ideas to test now. And I think one thing that the FDA could do would be this issue of so-called phase 0 testing, where cells can be microdosed and tested to see, do they hit the target and so on. And the issue has been, the main expense in a cell therapy trial is often a lentiviral vector or the cell manufacturing. But if we could test with GMP-like reagents in small amounts to ask, basic PK issues, even after direct administration to the tumor, does it hit the target? That could be a very useful way to speed in this phase 0 approach. So I think the FDA needs to adapt some of their guidance there for cell and gene therapies.
Marcela, you care to comment? One of the leading translational investigators at MGB.
Thanks. I was thinking more of a question of how do we compete on a global stage? I mean, you and I go back a couple decades now, and it used to be that we were the only ones doing this sort of thing, and by we I meant the U.S. But now you see this explosive growth, particularly in China, but also in academics and industry and biotech. I'm just wondering, how do you think about how do we, as Americans first, how do we keep our leadership, and how do we square innovation with safety and regulatory?
I think that's a great question. And you could all in the audience, if you type clinicaltrials.gov and then make your search term CAR T cell, end quote, and then there's a geographic tab. When I last did this, there's more trials in China than there are in the US or North America. And so part of that is due to prioritization within China and larger population base. The part of it is, I think, that their regulatory environment, they've made it easier to do early stage trials, which is what I think what we need. where the innovation needs to be to solve this problem in solid tumors. So in China, a single hospital trial can be done without the full GMP regulation. They require local IRB approval. And then if they want to move beyond that, then they have to do full INDs, and then very similar to what we would have with our health authorities here in the US. So I think if we had, I mean, they designed that from first principles in China because they didn't have the legacy of the drug development approaches we do here. So where they designed one more specific for cell and gene therapy needs, which are really to look at, deeply at small patient numbers with new ideas, and then, either kill it early as a bad idea that doesn't work, or go ahead forward. So the main issue is really cost of goods and time to testing. There's hundreds of ideas out there now to test. And right now, if we use a standard pharma model and do them all in serial, we're a long way away, I think, until we get a final solution for what we need to do to solve these difficult issues in solid tumors.
So we have about 5 more minutes left, and Marcela, if I'm going to ask you a question, you can't ask Carl a question, so you're going to have to answer a question here. So I'm going to ask you a question on toxicity. One of the questions from the audience: of the common adverse reactions to CAR T therapies, neurotoxicities, cytokine storm, which do you think is the most important to overcome to help more therapies advance through the clinic?
Well, cytokine release syndrome and the neurotoxicity, that's the transient kind that seems to be related to cytokines, the immune effector cell associated neurotoxicity syndrome. I think they're both important, and I think that they're both tractable. There's a number of trials now testing prophylactic strategies, combinations with cytokine blockade, earlier use of steroids. I think this is the kind of thing that needs to be solved with early phase clinical trials or small clinical trials. The incidence of CRS and ICANS are both high enough that you can treat 10 to 20 patients and with a potential prophylactic strategy, and then identify whether you have a signal or not. I would say, we initially started testing anakinra, as did a group at Sloan Kettering, and there seems to be some signal, but it's perhaps the dosing and schedule that needs to be optimized to really get that home run. But there are other possibilities out there as well. There's potentially blocking interferon gamma. There's earlier use of steroids, and Kite recently updated their label to use earlier doses of steroids. So I think these are solvable clinically with our clinical investigator group. Some of it could be solved at the CAR T sort of engineering and manufacturing stage. So, making gene knockouts for a CAR that doesn't have GM-CSF or a CAR that doesn't make interferon gamma are both strategies that have been published in vitro and in mouse models, but haven't yet been tested clinically, as far as I know. So it has potential to be changed at the CAR T design stage, but I don't also think that it's preventing patients right now from receiving this kind of care in tertiary academic medical centers. It is definitely a barrier for more accessibility and wider community usage. And I think if we figure out either a prophylactic strategy or a new CAR design to make it an outpatient procedure that is, that doesn't have associated toxicities that may or may not require intensive care, I think that's really going to be the next phase of sort of log growth, not just in the innovation aspect, but in the sort of uptake and accessibility aspect.
Great. Carl, I'm going to ask you a different question, if you don't mind. I know we just have a few minutes, and that is, in preparation for this, you mentioned some exciting data outside of oncology, and I thought maybe you should share with us what you are seeing in the CAR T therapies outside of oncology.
Yeah, so I'm really excited that, engineering cells, you can tune up and tune down the immune system. And York Set visited Penn about 3 weeks ago, and they had a case report of an Asian woman who had refractory lupus, systemic lupus erythematosus, in the New England Journal as a case report, a single case, about a year ago. And that caught my eye, because what they used was the same CAR that was made in my lab that's now Kymriah, that's used for lymphoma and leukemia. So they used this CAR that targets CD19 to target the pathogenic B cells that make double-stranded antibodies to DNA in lupus. And they found really a remarkable result, which was a complete response within weeks, as far as clinically with proteinuria clearing and the double-stranded antibodies going to undetectable. And that's now persisted for more than a year in that patient. And they have an ongoing phase 1 trial. This isn't Erlangen, Germany. And what the unpublished data is that 4 out of 5 patients have had complete responses. So in all my career for autoimmune disease, we've never had a curative therapy. You know, it's been TNF blockade, which is much better than steroids. But I'm optimistic now that some of these refractory rheumatologic disorders such as scleroderma, lupus, MS may be treatable with engineered lymphocytes.
Terrific. Marcela, any last thoughts or comments?
Yeah, no, I think, oncology is big, but it's also the beginning. I think I'm very excited about the potential in autoimmune disease, both in killing the pathogenic autoimmune cells and also perhaps just suppressing them with regulatory T cells. The immune system is so complex, and I think if we figure out how to hack it right, you can do so much in so many different diseases. Organ transplantation, autoimmune disease, fibrosis of various kinds. I think this is really the beginning in terms of autoimmune and other diseases. And not to mention, of course, regenerative medicine. You can do a lot with a stem cell, whether it's a hematopoietic stem cell or an induced pluripotent stem cell. And so if we can hack that and genetically modify, redirect, and infuse, and I think, obviously agree with Carl that we can do this in early phase 0 or phase 1 studies, we can get a very high signal, and then we'll know whether we're on the right track.
Terrific. So with that, I want to thank Carl and Marcela for this. We're going to change. A few people are going to come on. Marcela's going to step off. But thank you both for this session. Really appreciate it.
Thank you.
Thank you.